When we talk about types of mutations, there are a number of ways you can break these down and look at and differentiate mutations. So the first one is by cell type. So mutations can occur in germline cells or somatic cells, which are your body cells. And that just means that, as a result of meiosis, the germline cells, so future sperm and egg cells, may carry that mutation, versus when you do mitosis, only a single body cell may be affected. So the kind of after effects of these two types of mutations would be in a germline mutation, all the cells of the future organism are going to carry that mutation. So these are the kind of mutations that can be passed on to future generations, and maybe if they're a beneficial mutation, they can become a wider part of the population genetics. But with a body cell mutation, you're going to have a cluster of cells that grow from that one mutant cell as the cell replicates itself. And so that will just create a region in the body with that mutation, which may or may not have wide-reaching or even visible effects. Another and more common way of looking at mutations is kind of at the level of nucleic acid affected. So that can be at the level of the DNA nucleotide or the chromosome level. And so DNA nucleotide mutations tend to happen during replication. They're replication errors, versus chromosome mutations I associate with crossing over and misalignments during the process of crossing over. We're going to look at both types of mutations, but let's start with the DNA nucleotide type. So these are called point mutations, and they can be differentiated into two types. So you can just see we're kind of just subgrouping and subgrouping and subgrouping here. So the substitution type is where a single nucleotide is changed out for another. And those can fall into three categories as well. And I've listed them from like worst to possibly, most harmless. So nonsense mutations create a stop codon. So that means when this DNA is translated into mRNA, where there was no stop codon because maybe it was in the middle of a protein coding region, now there's a stop codon, and that protein is just much shorter than it would have been. A missense mutation just puts a new amino acid in place of an old amino acid, a different amino acid. And so this can be pretty catastrophic to the shape of the protein, or not very much at all, because if you change, a positively charged amino acid for another positively charged amino acid, that's not going to cause much effect to the overall shape of the protein. But, a polar amino acid turning into a non-polar amino acid, or the other way around, can have a big effect on how that protein folds, because non-polar amino acids are going to be clustered together by mutual exclusion, and hydrophilic things are going to pull to the outside of the protein and face the water. So that can be a big deal. Silent mutations change the codon in such a way, and again, we're talking from DNA to mRNA, now the codon is changed, but you get the same amino acid because there's so much redundancy in our codon language, and so that can happen as well. On the frameshift mutation side, you can have insertions or deletions. So both of these are going to cause a shift in the reading frame of the codon, and that's why we group them together under frameshift. So here's an example of, and I swapped them, my deletion and my insertion. Let's just take this, move it over here. That'll test how much attention y'all are paying. All right, so insertions and deletions. So it shifts the reading frame, right? So where at the beginning, say these were two codons and that was the way they were read in sequence, now the language completely changes. Imagine taking your favorite line from a book and taking all the spaces and moving them back or forward one letter. It's going to change the meaning drastically or make it make no sense at all. So frameshift mutations can have a big effect, especially if they occur early in a protein coding region, but even later in the protein coding region, you could change where the stop codon falls or have no stop codon at all. So big impact. Frameshift mutations have big impacts. Now we're going to look at the chromosome level. So here is a chromosome, and I've just labeled some regions so that we can track them. And so one thing that could happen is if you have a cross and like a misalignment, then one region can end up being deleted. So you can see the misalignment here where D should have been beside D. And so this area was deleted, and we get a copy of the D and E regions in this area. So sometimes this can be a really cool thing. You know, it can create new copies of the gene that evolution can now start to work on, because mutations could accumulate in one D region and you still have another functioning D region. So all the genes in this region continue to do their job, and maybe in another region, mutations can accumulate. So we see gene families come out of these type of mutations. But overall, you can have big dosage impacts when you're making twice as much, because if you turn on a gene in region D, you're going to get twice as much protein from that change. So that's a big deal. And obviously, not having the genes at all is a big deal. Another type is an inversion. So we see this when the chromosome kind of curls around itself and crosses over. You can get, and I purposefully left the letters upside down, obviously, you know, there are no letters labeling actual chromosomes, but we do have that 5' to 3' orientation in those antiparallel regions. So we've also flipped that orientation whenever we have an inversion. So that's something to keep in mind. The last chromosomal mutation we'll look at is a translocation, and that's where crossing over occurs between two non-homologous chromosomes. So say this is chromosome 3 and 5 or something like that, and the regions actually swap here. So we have a bit of chromosome 5 on chromosome 3 and a bit of chromosome 3 on chromosome 5. When you look at major transitions in life, so related organisms, and you look at their chromosomes, sometimes you can see evidence of this in the banding patterns. So it does cause an effect. You don't have any additional chromosome or additional copies of your genes, but they are located in a different place. And if you take this to an extreme, you could, you know, if Q crossed with K, you could take two shorter chromosomes and create one gigantic mashup chromosome. And we actually do see that in our chromosomes. Chromosome number 2, I believe, is a fusion from our ancestral organisms that might have had more pairs of chromosomes, 24 pairs instead of 23 like we do. So something to think about, lots to chew on here, but that is mutations in as much of a nutshell as I can put them in.